Thank you. This is a good example of how scientists move around or can be moved around. It turns out I'm now an emeritus professor from the Clare Hall laboratories in London. So now I'm instead at the Francis Crick Institute, which hasn't opened yet. So I'm an emeritus in a place that's not yet open. I'm going to talk about DNA. And as you know, DNA is the carrier of genetic information. And it has been assumed because of that DNA must be an extremely stable molecule. Any geneticist would tell you that a change in the DNA structure might lead to mutations or cell death. And it was then supposed that DNA must be very stable. But the components of DNA are nucleotides, common building stones. There is nothing unusual about them. They are not any more or less stable than most amino acids are. And so there is something here that doesn't quite make sense. You think of DNA like this. You see it in an instrument park, a stainless steel structure, but that's not the way it is in the cell. And so there is a dilemma here. And the answer to try to study that problem, I many years ago got interested in how labile is DNA. Can we measure its lability? Oops. And the way you do this is by classical biochemistry. The simplest way of studying the lability of DNA is to look at the individual building stones in DNA by itself. So you make DNA from microorganisms where you can get mutations, E. coli or Bacillus subtilis. So you can study the fate of a component of DNA by itself. For example, oops. It's very sensitive, this. So you can make DNA, for example, labeled just in cytosine residues, and then you incubate that DNA for long periods of time at different temperatures, and that gives you an idea of how unstable that component of DNA is by itself. You then have to hydrolyze your DNA with reagents, enzymes, and by chromatography separate the components and see what has happened to the cytosine. So in this case, for example, you will almost immediately find that cytosine then is degraded to uracil in DNA. And there are several susceptible sites in DNA. There's not just one site of damage, but there are many sites of damage. One of the more important ones, or perhaps the most important, are the base sugar bonds. This bond here, and also this bond here. And if that is broken, you get a loss of base in DNA, which obviously leads to loss of genetic information locally. And, but it doesn't break the DNA chain by itself. You just have a gap where there should be a piece of information. And to study this, we have labeled DNA in different ways and studied the components. And I have made little arrows here for the thing. Little arrows where you can see the main targets in DNA, and there are many of them. And because there are many targets, there are many changes in DNA. Some of them are caused by water, and I'll come back to that point. Others are caused by oxidation or by reaction with small molecules in the cells. So there's quite a zoological garden here of different DNA lesions that can arise under in vivo conditions without any external provocation, such as radiation. And since we could label DNA specifically, we could then also find out how fast are these reactions of DNA decomposition. And here are several of them shown. I think I need some help with this one. Can you take over this? If I say next slide, you save a lot of time. Yeah. Yeah, back one. Go back again. Yeah, that's fine. So here are several of the... Okay, now I need this to have a pointer, right? Yeah. It's not very well organized, this. There are several thousands of purine bases lost from DNA in a single mammalian cell in 1 day. And it's fairly obvious that that will lead to such a massive loss of genetic information that the cell has to do something about it. If you have lost a base in DNA, the conclusion is that you really have to repair it somehow. And the way you can repair it is to read information in the complementary strand of DNA. So that's why it's so important that DNA is double-stranded. If you lose information on one side, you can always use the other strand to find out what should be the correct information. And this is a so-called excision repair process. Here you have lost the base in DNA, and to replace that, you cleave the DNA chain and take away the sugar phosphate residue that was there before, and then you use the DNA polymerase for short patch gap filling, and finally a DNA ligase to rejoin the chain. So that's the model how you could repair this. And if you have a damaged base, there is also a separate enzyme which takes out the damaged base, in this case uracil, to leave an abasic site, and then we are back to the repair pathway I described. And we have purified these enzymes and reconstituted the reaction with purified enzymes, as biochemists like to do. And this is shown over here. You make an oligonucleotide of 41 residues long, label it on one end with P32. In the middle of this is a uracil residue where there should be a cytosine residue. And then you anneal this to a complementary oligonucleotide and expose it to these various enzymes stepwise. And this way you can reconstitute a pathway. So here you can show that the DNA molecule, it retains its length once the uracil has been removed. But then a second enzyme comes along and cleaves the DNA chain at the site of base loss. And at that site, then there is DNA synthesis and ligation. So you're back to the normal pathway. So this piece of DNA is now identical to this piece of DNA. And by this excision repair system, you have repaired the DNA. And this happens all the time. As I said, 10,000 times a day, at least in every cell. So it means DNA is not totally stable, it slowly turns over. There's also oxidative damage to DNA. And the main point here is that the damage you get with oxygen is quite different from what you get due to water. So these are new problems for the cell, and you have to evolve new repair enzymes to deal with oxidative DNA damage. Particularly dangerous one here is oxidation of guanine in this position to 8-oxoguanine. Which changes the coding specificity in DNA because the base flips around 180 degrees in DNA. So it turns itself upside down, and then it will base pair with a purine instead of a pyrimidine. So you get the so-called transversion mutation. And this is a highly mutagenic change the cell has to do something about. And there's also damage due to several small molecules in cells, like S-adenosylmethionine is a coenzyme in cell, and it's the preferred methyl group donor. This methyl group is easily donated in many transmethylation reactions, and it spontaneously can also be released and react with molecules. And there are many sites in DNA that are susceptible to such alkylation by S-adenosylmethionine or methylation. And this is a completely different kind of methylation from biological methylation of cytosine to 5-methylcytosine, which changes the signaling system involved. This methylation I'm talking about here is just DNA damage, and it should be removed as quickly as possible. So the methylation, the alkylation of DNA turned out, again, to be fascinating to a biochemist because you can remove a methylated base from DNA, like 3-methyladenine. Here's a methyl group in the minor groove of DNA where methyl groups don't belong. It interferes with transcription or replication of DNA, so this is a lethal change, and it has to be removed, and it's removed stepwise. In very much the same way as you deal with an abasic site in DNA. I should also mention there are occasional lesions in DNA, like 7-methylguanine, where the lesion is not dangerous. It's not miscoding, and it's not lethal to the cell. So it's just tolerated and slowly diluted away. But that's the exception, and most DNA changes require some active repair in the cell. So this is a standard excision repair, but then there are other interesting lesions like this 0,6-methylguanine, which can be removed very quickly just by demethylation. A special enzyme will remove the methyl group from DNA and transfer it to a cysteine residue in a protein instead. And this is work done by Monica Olsson, who is in the audience here in my lab when I was working in Gothenburg several years ago. And so what do you do now? Now you have the repair protein has got a blocked cysteine residue. So the initial reaction is you should remove this methyl group, but that's not so easily done because methylcysteine is a very stable compound. You can boil it in hydrochloric acid and nothing happens. So the cell ignores the problem, it just sacrifices this protein. So it takes one repair molecule here to remove one lesion from DNA, and then that has been used up, it can't be used again. And now the nomenclature experts get upset and say, can you call this an enzyme if it only acts once? Perhaps not, but it acts very much like an enzyme deals with the so-called suicide inhibitor. So this is a natural case of that. There are also other interesting forms of DNA repair of methylated DNA. You can get toxic methyl groups here on 1-methyladenine, on 3-methylcytosine. They interfere with base pairing and can't be tolerated, so you have to remove these. So we know that there was a gene for this called ALK-A, or ALK-B rather, in mammalian cells. And we could clone that gene, express the protein in large amounts, and mix it with damaged DNA, and nothing happened. And we stayed at that stage for a long time until we finally, after various sequence studies and so on, found out that this enzyme has unusual cofactors. It requires ferrous ions and also alpha-ketoglutarate. So if you don't have those two components in your reaction mix, nothing will happen. But once you add them, the repair reaction takes place. Mm-hmm. So this is as far we got. So here are some of the components in cells that will damage DNA without external influence. There's water. Water is a group-specific reagent, and it's a very weak one, so you think you can ignore it, but you have to keep in mind that in the cell, there is a lot of water all the time, so slow hydrolysis of DNA components can't be avoided. And there's really no way you can protect your DNA against water. It's, the only way you can do is repair it. There's also reactive oxygen, and as I mentioned, there is several small reactive molecules, like S-adenosylmethionine. In some reactions, formaldehyde is generated as a side product, and that will damage DNA again in new forms of base damage, which makes it necessary to develop new kinds of DNA repair enzymes. And this has been studied by K.J. Patel in Cambridge recently. And if you don't have that kind of repair, you get severe damage to the DNA. So the diversity of lesions means that there has to be a large diversity of repair enzymes. And I think we are so far more or less scraping the surface here, because there are many DNA lesions in cells. Many of them caused, for example, by reaction with metabolites or with coenzymes, and this has not yet been studied. If DNA is damaged by a coenzyme, like this one, or some metabolite like formaldehyde, you have to have a repair system for it. And those experiments can be done, but first one has to make studies with damaged DNA, and so you know what you're looking at. And my prediction is that there will be several more repair enzymes acting on interesting forms of DNA damage, which haven't been studied yet because the lesions haven't been characterized properly yet. I think that is all I'm going to say about DNA repair of spontaneous damage, but there are other forms of DNA alterations in cells that you have to be aware of. and especially enzymes that act at strand breaks, and the typical ones are so-called exonucleases, an enzyme that will act at a strand break and start degrading the DNA from that point. And they have key roles in recombination, in DNA replication and repair, but they obviously have to be controlled. So if you look at what kind of enzymes do you find of this type in mammalian cells, there are two key players. There is one called FEN1, or originally DNase 4, and it removes protruding ends or flap structures or damage at 5' ends. And I won't have time to talk about that in any review article on DNA replication, you will find described what the role of the important role of FEN1 is now in DNA replication. There is another enzyme that's abundant in mammalian cells, and that has been studied much less because it's not present in yeast or E. coli or, well, not only yeast, but other lower eukaryotes. It has about the same distribution as the immune system, so it's specific to higher cells, such as mammalian cells. And what that one does, its key function is to just degrade DNA from the 3' end, and it does so very effectively remove any kind of single-stranded DNA. So why is that there? First, let me introduce it quickly. It's a small enzyme. It's a single exon enzyme, which has made it very much easier to study it in human disease because you don't have to make cDNAs. You can just sequence the DNA from the patient directly to see if there's a mutation there or not. And it's a small enzyme, very high turnover number. And like most exonucleases, it has magnesium binding sites in it. This is different from other exonucleases in that it also has a long hydrophobic C-terminal tail of some close to 100 amino acids. And we don't know what this DNA, where it comes from or what it resembles. There's nothing in the database that looks like this DNA. And how this evolved, we don't know. When we looked with antibodies where this enzyme is in cells, we had a bit of a surprise because we know it was in or with the cell nuclei, but it turns out in growing cells, it's actually distributed like this. You can see down here, it's the same distribution as the endoplasmic reticulum. In a way, that doesn't make too much sense. Why would you have an exonuclease in the endoplasmic reticulum where there is no DNA? But the answer to that is that a small part of the enzyme is in the nucleus, but perhaps more importantly, it can be mobilized if it's needed. So that this is where you usually see this enzyme. If you now give the cell a dose of ionizing radiation, the enzyme is mobilized and instead goes to the sites of chain breaks in DNA. And when the chain breaks have been repaired, it returns to this position. We know very little about how this is regulated. When I started in biochemistry, we thought of the cell as a bag of enzymes. You broke the bag and solubilized the enzymes and studied their biochemical properties. But there's clearly a lot of site-specific effects here. What keeps this enzyme in place? What mobilizes after DNA damage, so it goes to the sites of damage? We just don't know. So I think this is another fascinating part of studies of these enzymes. What are the signals that can mobilize it? Okay, so I've said something about TREX1, and all right, perhaps nuclease doesn't sound like such a fascinating enzyme, so is it important? And to find out, you do a knockout mice first, and you are lucky if your experiment works, because if you knock out the gene, or those of you who have done this game know that what sometimes happens, you get no phenotype, and then you're not much wiser, or the cell dies, which means that the gene you're studying is important, but you don't know why it's important. But in cases like this, you can also get better information, because here the cells survive, but as you can see, they have very strongly reduced aging. So this is knockout mice, normal mice. And this is the lifespan of mice where you have knocked out this enzyme. So they have clearly strongly reduced lifespan. And why is that? Well, you don't have to be a master pathologist to find out. You take the mice with this lesion in it and study them, and it turns out that this is a normal heart, and this is what the heart looks like in a knockout mouse. Massive myocarditis, and so these are T cells infiltrating the heart muscle, and what ultimately often happens is that the animal dies from heart rupture. I had some control with the laymen that look into danger of animal experiments. And they were upset with me because they really thought it was very terrible that the mouse died from a broken heart. So I shouldn't do experiments like that. But this is the broken heart is just due to a weakening of the heart muscle. And then it turns out there is a human disease called Aicardi-Gouttiere syndrome, which has the same phenotype pretty much as these mice, with a heart lesion and so on. And it's an autoimmune disease, which makes it interesting because it's an autoinflammatory neurological disease, I should say. And it has some resemblance, or many resemblances, to systemic lupus, which makes it interesting because that means that this TREX enzyme might be an important key to understand the lupus erythematosus and diseases of this type. And you get this phenotype from the loss of TREX1, and it turns out another lesion in cells, which is the RNase H2, which is the enzyme that removes RNA primers during lagging strand DNA synthesis, also has this phenotype. And there is an explanation for that I'll come to in a minute. So if this enzyme, which removes single-stranded DNA, is deleted, to what extent is the phenotype due to accumulation of single-stranded DNA in cells? Well, to a large extent, actually. First we have to show that single-stranded DNA did accumulate, and I don't have time to go through the details of this protocol, but we can isolate single-stranded DNA from cells, and this is what it looks like. So this hasn't been seen before, because in normal cell that has the TREX enzyme, the TREX1 enzyme is very efficient and degrades single-stranded DNA fragments very quickly. So you just don't see anything. Oops. But here you have accumulation of DNA fragments of between 40 and 80 nucleotides long of single-stranded DNA in the cell nucleus or in the cell. And that turns out to have a damaging effect. This is just to show that it is single-stranded DNA we are looking at. And here is a model for what happens in this generation of single-stranded DNA. It's a side product of lagging strand DNA synthesis. And so when you synthesize a lagging strand of DNA, you use DNA polymerase alpha to make a small primer, and then you make a longer piece of DNA, which is the bulk of the lagging strand, up to the next fragment, the so-called Okazaki fragment. And then you have a special enzyme, RNase H, to remove this RNA primer. And you then use DNA ligase to join the lagging strand of DNA. And this is a very streamlined procedure because all our DNA is susceptible to this, uses this method for lagging strand synthesis. Occasionally, there is something goes wrong in that the polymerase is very efficient and it can displace the primer strand like this when the DNA breeds. And then you have the FEN1 enzyme I mentioned in passing before to remove this overhang structure so that you can still ligate the DNA. Yeah? And to do this, to repair the DNA, you have to remove this RNA primer from DNA. So the special enzyme RNase H2 that removes the primer here is important for DNA replication, and if you don't have it, this kind of event will be much more common. And we think that explains why the phenotype of an RNase H2 mutant is very similar to the phenotype of a cell where single-stranded DNA fragments have been generated. If you don't have TREX1 enzyme, this DNA again is a negative component. The reason that it is a problem is due to the immune system, but let me show you here. The first question when we saw this single-stranded DNA fragment, does it represent all DNA or is it some subclass of DNA? And there was an American group that also took an interest in this and felt that perhaps this single-strand DNA was retroviral DNA or some other subclass of DNA. Well, I don't think that's right, and with the help of colleagues in Leipzig who are experts on dealing with tiny amounts of single-stranded old DNA, we could show that this DNA actually, as you might expect, if something goes wrong, generally lagging strand synthesis represents all DNA sequences. There's no enrichment of any specific form of DNA there. So you have the problem here that there's a piece of DNA here that's up to 80 nucleotides long, and it turns out in mammalian cells that's long enough to trigger the endogenous immune response. It's triggered by oligonucleotides that are over 35 nucleotides long. Yeah? So this means you switch on an immune response here. And we think that explains the main phenotype of the human disease where you lack this TREX1 enzyme. You have massive production of interferon alpha, and that is associated with an autoimmune response. So those patients are severely ill and die at young age. So that's just a little bit of a reminder of that DNA damage can have several aspects to it. It can be just due to the lability of DNA itself, or you can have enzymes in the cell that generate side products of repair reactions that also are deleterious to the cell, and in this particular case switches on the autoimmune response. So while DNA repair for many years was thought to be a rather small form of correction mechanism, we find now there are more and more aspects of it, and it overlaps here with autoimmunity and with other interesting human diseases. So I'll stop at that note, I think.
Thank you, Tomas, for a very inspiring lecture, going from history into the future of DNA repair. We have time for questions, so yes.
Thank you. This intrinsic fragility in the DNA.
Can you repeat the question?
Is it the same with RNA? And in that case, how does the body, and also more interestingly, perhaps retroviruses, treat the RNA that breaks?
Yes. Is there a corresponding system for RNA, keeping up the RNA stability?
No, there isn't really, because it's easier to make a new RNA molecule. But it's much more difficult to make a new chromosome. So you have to be able to repair your DNA, but it's not so important for RNA.
On the other hand, though, there is no failsafe in the single-stranded RNA molecule, whilst there is in the DNA, right? Or
that's another reason why you can't do it. There is not an opposite strand which you could piggyback to know the right.
That's right.
answer.
Thank you.
I should say there are repair systems where you don't use the complementarity of DNA. If you have a double-strand break in DNA, it can be repaired in a sophisticated way by recombination, so the damage is introduced. But under more desperate situations, like high doses of ionizing radiation, the cell can just join the two blood cells together. And that's a very risky business, because the two ends might come, it's not necessarily, when there are a lot of broken DNA molecules, there's no mechanism to make sure that you join the right ends to each other. And this is a common way of introducing chromosome aberrations.
That's very interesting, thank you.
There is actually a future maybe fictionless application of the machinery that repairs double-strand breaks, which may be called gene correction therapy. If you want to change a gene, you could, and Aaron Klug has been involved with that.
It's a very active research field now.
Yeah. So you can, using, for instance, zinc fingers, identify the place where you want to create a double-strand break. The question is then, how does the machinery include the oligonucleotides you add to the patients? How does it, you need high concentrations or?
Well, I think the problem is more when you introduce a strand break, it's often due to a chemical reason. There might be some damage around the site or breakage. And you can use the information in the other chromosome in the cell, but if you repair it by such, there is a risk that you just introduce errors. Now, most of our DNA is not coding DNA, and even within the coding regions, some of the DNA introns and so on are not important. So you would be unlucky if you just introduce one DNA error and that has a mutagenic effect.
Yes.
Yeah. I have a question. It's in one of your early slides, you said I interpreted it as it's more common for depurination than the pyrimidation. Why is that so?
The pyrimidation was more common than the pyrimidation. That's the question?
Yeah, exactly.
Yeah, the purine repair was more efficient.
It's more, yeah.
It's more common. Okay.
It's more common, yes.
Yeah. So how come?
Because the base sugar bond that keeps the purine in DNA is the weakest link in DNA. So that's more commonly broken. And then the base just is fed to the hungry tiger somewhere and disappears. And you have to replace it. But the strategy for replacing it, the excision repair mechanism, can be applied to any kind of DNA lesion. You just have to have a mechanism to initiate the repair.
Something that struck me as it had to do with aging was your diagram about the mice, the healthy mice who died suddenly, and those with a broken heart. That showed an exponential, something like that. Do you have an explanation why it looked like that?
I think in an immune deficiency disease, there are always going to be some organisms that are more resistant than others. So I'm not surprised. There are many diseases that are common or dangerous in childhood. There are some long-term survivors often. A small minority.
Yeah. So those normal mice, it was more a programmed death, whereas the other had more a stochastic.
I don't think it's programmed death. It just the immune response was triggered much more rapidly there to cause damage.
Okay. Yeah. Can you speak closer to the microphone?
No increase in tumor formation in your knockout mouse, your TREX1. Can you comment on why mutations in certain DNA repair enzymes do show an increase and why others do not, or do we know much about that?
Could you try to repeat the question?
Yeah, I was wondering why there was no increase in tumor formation in the TREX1 knockout mice. And I'm wondering, why do mutations in certain DNA repair enzymes cause an increase in tumor formations while others maybe do not?
I'm not sure
Well, I didn't hear it well, so...
You didn't hear it at all?
The frequency of the tumor...
Yeah, so a knockout of TREX1.
Knockout the TREX1.
Yeah, there was no increase in spontaneous tumor formation in those mice.
No increase in spontaneous tumor formation.
Right.
That is true. Of course, then you have to think of that these animals die young, and a limited amount of animals have been studied. So if there was a slow tendency to increase carcinogenesis in these mice, we wouldn't have detected it perhaps. But there's no dramatic general overall in carcinogenesis in the mice.
Any more questions? People long for lunch. So we thank you again.
Thank you.